Thank you. Yes, so this is a project update on kind of the parallel project to what Aaron just presented on. So I get the luxury of being able to breeze through some of the introduction because it's much of the same motivation and the reason that we're doing this work. So this is just looking at whitefish tagged in U.S. waters. So this is a massive research team that we have put together here, many folks in the room, and just want to acknowledge all the work that they and many others have put in, and especially the field crews that have been tending these acoustic receivers. This work wouldn't be possible without them, and this work was funded by the Great Lakes Fishery Commission. So many of us know the Lake Huron and beyond whitefish story, and that's that we're having ongoing declines in recruitment and commercial fishery yield, notably over the last couple of decades. So there was this peak, at least in Lake Huron, in fishery yield around the year 2000, and we've had declines since then. And these declines have coincided with major ecosystem changes, including the expansion of zebra and quagga mussels, the invasion and expansion of round gobies, and the collapse of the important prey species Diparia. And we really have a lack of fishery independent movement data at the whole lake scale in Lake Huron. We have some mark recapture data that utilized returns from the commercial fishery, but that was done about 20 years ago. So now we're looking for new information on how the fish are behaving today. Because right now we have knowledge gaps in the stock structure and how these stocks are contributing to the different fisheries, as Aaron talked about the different management zones that we have throughout Lake Huron. So this leads to our research questions, some of which I'll touch on today and some of which I'll save for a later date. But our overarching goal is trying to understand the potential for mixing among spawning populations, understanding source sink dynamics, so understanding where fish might be being produced more than they're harvested, or vice versa, where they're harvested more than they're producing, understanding the overall population structure, and then the movements of lake whitefish generally and how they've changed over time. So this is now our third acoustic telemetry talk in a row, so I don't need to spend much time here. Hopefully you're not tired of these talks yet. But we tagged these fish at a variety of locations, and then they were heard throughout the Lake Huron receiver array. And as you can see from these pictures, we tagged in a variety of conditions in October in northern Michigan. It was either 40 degrees or 60 degrees and didn't seem to be in between. So this map here shows all the different receiver locations with the black points where fish could have been detected. And then the colored points show all the different tagging locations in U.S. waters. So we started tagging in 2022 and just wrapped up our tagging last year. And we tagged across three general regions, up in the north on the UP shoreline, in kind of the central part of the lake around the port of Alpena, and then a couple locations in Saginaw Bay. And so an important distinction between what we've done on the U.S. side and what Aaron just presented is that we were tagging fish out of commercial trap nets pre-spawn. So these were mostly late October, some just in the beginning of November. So we make an assumption that these fish were roughly in the region that they might be spawning, but we certainly weren't picking fish up off of spawning reefs like the Canadian folks were. So in U.S. waters, we've tagged just under 800 lake whitefish, which brings the total across Lake Huron to just under, I think, right around 1,400. So a lot of lake whitefish with acoustic tags swimming around out there. So today I'll be talking about apparent fate of these fish, overall space use, management unit occupancy, and then finishing with some early work in genetic population structure. So much of the same kind of stuff that we just saw from Aaron. And I just want to highlight now, and I'll probably mention it again, that this is just a project update. We're still collecting data from these fish. Many of these fish still have many years of data or battery life left in their tags. So this is just scratching the surface of what we hope to present on this data. So I'll start out with just mentioning the fate of some of these tags. So these are 3+ year battery life tags. So hopefully by the end of this project, we'll get multiple years of data from many of these fish. And as Aaron was just talking about, there are different sources of shortened lifespan that we can see in these tags. including tag returns from harvest, constant pings on a receiver where we assume that that fish either died of some sort of natural cause or maybe lost its tag, or fish that go missing out in this array, which we think if fish are missing for more than about half a year, they're probably either dead somewhere or were harvested and not reported. So we don't know the fate of those fish. So we saw right off the bat within the first 30 days that about 16% of our fish died from some cause or another, which for a Corrigonian tagging study we think is fairly good. These fish are thought to be pretty sensitive to the tagging process, so we were pretty happy with this number. And after 1 year, we saw that about 50% of our tags are still alive and active. So this will be a focus of future research. I don't go into any more detail now at this time, but as we collect more years of data, we really plan to dig into potential causes of mortality and different spatial structuring of that mortality. So now I'm going to step through some plots that are similar to what Aaron showed. So these are just detection bubble plots. So all the X's show locations where receivers were that fish were never detected. The star shows the tagging location for this tagging group, Hessel. And then anytime there's a colored bubble, that means that at least one fish was detected there. And if you could focus on the areas where there are darker, the more purple and blue colors, that's where the majority of the tagging group was detected. These yellow colors are where only a couple individuals were detected. So here for this Hessel tagging group, we can see that the majority of these fish are staying quite local to their tagging locations. So they're staying up there in Northwest Main Basin, some of them coming up into Pottaginnasing Bay and just the edge of the North Channel. And there are a couple individuals that do move down into the southern part of the lake, but that's only 1 or 2 fish. These detour tagged fish show a similar pattern, but they're even more local than the Hessel fish are. A lot more fish tagged in detour. You can see that the scales on each of these maps is different based on the number of fish that we tagged at that location. But those fish tagged at detour are remaining really local to that detour area. Some of them moving into Pottaginnasing Bay in the winter and spring, but a lot of them staying just right there in Detour Passage and that vicinity. Moving down to the more southern part of the lake, we see this central Michigan tagging location, which I combined Alpena and North Point, which were very close and showed similar behavior. They show very different movement patterns, right? These fish are spreading throughout the lake to the south. Many of them are hanging out in kind of that area between Thunder Bay and Saginaw Bay, and some are even moving into the southern Main Basin, which is quite a large movement for these fish. And what's really interesting, I think, is that we tagged a good number of fish here, and only 1 or 2 moved north of where their tagging location was, which is notable because mark recapture work done in the early 2000s showed that fish were showing these kind of broad scale movement patterns, but they were dispersing to the north and to the south in that previous study, where here we're just seeing fish disperse to the south. And then fish tagged in Saginaw Bay, both around Tawas and around Caseville, showed similar broad scale occupancy patterns to those fish tagged around Thunder Bay. And that's that many of them were hanging out in either the Saginaw Bay area or the southern Main Basin. And it doesn't really show up here, but fish seem to show variable movement behaviors that they were either hanging out in outer Saginaw Bay or they were moving down to the southern Main Basin. And we haven't looked in great detail yet, but those behaviors seem to be somewhat repeatable. So shifting gears a little bit here, these plots that I'll show over the next couple of slides show the proportion of fish from each of these tagging groups that were detected in any management units. So the management units here are colored, and those correspond to the colors on the bars. So this first example here, and this is broken down seasonally. So this first example here from the Hessel, during winter, we can see that about half of the fish tagged in Hessel were hanging out in management unit 1. That's where they were tagged, with the other half kind of spreading out between management units 2, 3, and the North Channel. So looking across the year for these Hesselfish, we see somewhat consistent patterns that these fish are spending, most of these fish are spending time in the management unit that they were tagged, with some moving over toward the detour area and up into Pontiac Bay. These detour fish, like we could have expected from those bubble plots that I showed previously, are really hanging out in that management unit. They were tagged management unit 2. But during winter and spring, about half of those fish are moving up into Pontiac Bay and hanging out in that adjacent management unit. But again, these two tagging groups are staying in this northwest area of the lake. Very different colors showing up for these central Michigan fish. So they're kind of spreading out, and each fish is doing its own thing, hanging out in either the central part of the lake, Saginaw Bay, or moving down to the southern Main Basin. And you get those darker colors from Management Unit 8 on the US side or 4-5 on the Canadian side. And then in Saginaw Bay, you see these same kind of colors that you see with the Central Michigan group, but in different proportions. So a lot more of these Saginaw Bay fish are staying within the Saginaw Bay Management Unit, but you do have some individuals that are moving down into the southern Main Basin, and some move slightly north to the adjacent Management Unit 6. And the last couple of results slides that I'll show here have to do with population structure. So we did rad-seq on all these fish that we tagged, and these plots here are PCAs that show the differentiation of the genetics of these fish. So the big takeaway here on this first slide is that there's very low overall genetic differentiation. So we have support through k-means clustering for 3 genetic clusters here. But as you can see, these 3 clusters are somewhat smeared together. They're not distinct groups and clusters. They're all kind of blending together. So this suggests that there is some strain going on and some hybridization going on between these clusters. They're not distinct population units. But what we do see, which is quite interesting, is some broad scale population structuring going on. So this plot here shows each bar on this plot is an individual fish, and the color corresponds to the cluster that it assigns with. These cluster numbers 1, 2, and 3 are just arbitrary numbers. So what we can see is that, and these individuals are broken down by the management unit that they were used most detected in during November. So this is different than the tagging location grouping that I've showed, and instead I'm using the detection data to tell us where these fish might have been spawning based on where they were in November. So the fish that were in management units 1, 2, and 3 during the month of November, almost all of them assigned to cluster 2. Whereas in the southern part of the lake, the fish that are using management units 5 and 6 are tending to assign to cluster 1 with some cluster 3 mixed in there. And vice versa for management unit 7, that about 70% of those individuals are assigning to cluster 3, with the remainder assigning to cluster 1. So we see this really strong divide going on here from essentially the north versus everybody else. And then within the more southern, the Saginaw Bay, Thunder Bay area, we see some spatial clustering, but it's a little messier. So overall, our takeaways so far are that we have highly variable movement behavior, both within and among our different tagging groups and regions of the lake. And there is some apparent broad-scale population structure, which aligns the genetic and behavioral separation that's going on with this more northern tagging groups from the remainder of the lake. Whereas in the Central and Saginaw Bay areas, these fish are showing some spatial structuring, but there is some overlap going on, both the genetics and the movement behavior. It's a little messier there. And we have some mixed stock fishery going on in the central and southern part of the lake, where you get contribution from both those fish that we tagged in the Thunder Bay area and the Saginaw Bay areas. And like I mentioned at the top, this project is very much ongoing, so we have years left of battery life on many of these tags. So we have lots more work to do here. Where we'll be looking at drivers of movement, behavioral group structure, trying to identify to whatever extent we can spawning groups and fidelity to those spawning locations. Like I mentioned, the mortality aspect, and then further getting into the genetic structure and potential for adaptation. And with that, thank you for your time and attention, and I'll take any questions.